So I'm able to run normally because I don't have osteoarthritis, but my friend Deej here does have osteoarthritis, which is why he had that little shuffle walk. Can't run as well. His joints don't work as well anymore. Right, Deej?
Every day, thousands of lives are affected by osteoarthritis.
Osteoarthritis is a disease caused by the wearing down of protective cartilage that covers bones, seen in joints, mainly the knee, hip, and hands.
Osteoarthritis affects many different parts of the body, including the wrist, the elbows, the shoulders, the pelvic regions, the knee, and the ankles.
Osteoarthritis can cause extreme discomfort and pain in those who have it. It can also lead to the growth of bone spurs, which can be only removed by surgery.
This is an animation of a knee. Over time, the cartilage wears down. This causes a space to form in between the bones. Every day, as this joint is used, bone spurs can form. These can cause very sharp pains and swelling in the joints.
Luckily, thanks to scientific advancements involving stem cells, a cure is in sight.
There's a cure!
Stem cell research is a study of using embryonic stem cells to produce more specialized cells.
Okay. In the early stages of development, unspecialized cells, also known as stem cells, can be tricked into forming new specialized cells such as organ or tissue cells.
Embryonic stem cell research has been causing some controversy in the world due to its close ties with abortion. In order for stem cells to be harvested for embryos, human fetuses must be destroyed in order to harvest the cells.
In order to avoid controversy, Johns Hopkins Medical Center started using stem cells from adult girls.
While other universities focus their research elsewhere, UCLA has been looking for a hormone that will help activate stem cells quicker once they're injected into patients.
These cells can then be differentiated into any type of mesenchymal cells.
These can be harvested and used to regrow the protective covering on joints of those with osteoarthritis.
The series entitled TED Ed recently made a video documenting the idea of stem cells and what they are.
Stem cells are cells that are undifferentiated, meaning they do not have a specific job or function. While skin cells protect your body, muscle cells contract, and nerve cells send signals, stem cells do not have any specific structures or functions. Stem cells do have the potential to become all other kinds of cells in your body. Your body uses stem cells to replace worn-out cells when they die. For example, you completely replace the lining of your intestines every 4 days. Stem cells beneath the lining of your intestines replace these cells as they wear out. Scientists hope that stem cells could be used to create a very special kind of personalized medicine, in which we could replace your own body parts with, well, your own body parts. Stem cell researchers are working hard to find ways in which to use stem cells to create new tissue to replace the parts of organs that are damaged by injury or disease. Using stem cells to replace damaged bodily tissue is called regenerative medicine.
Cells have shown great promise in the science community and are one of the most promising new ideas of modern biology.
We were lucky enough to have the opportunity to go inside Rutgers University's biomedical research facility and observe and interview medical professionals.
Inside the lab at Rutgers Biomedical Department under Dr. Yarmush, we met with a grad student who has had the opportunity to study the use of mesenchymal cells, also called MSCs, to cure osteoarthritis.
Hello, my name is Juliana Majero-Rios, and I'm a fifth-year PhD student in biomedical engineering at Rutgers University. I did my undergrad in microbiology and biotechnology in the University of Puerto Rico at Maya West, and I came to Rutgers to pursue my PhD as a biomedical engineer to study how we could use mesenchymal stromal cells, or MSCs, to treat osteoarthritis. So on a day-to-day basis, I work with tissue culture, and I culture the mesenchymal stromal cells, or MSCs, and so I work with tissue culture, and and my project specifically looks into using mesenchymal stromal cells in alginate capsules. So alginate is a polysaccharide that can be extracted from algae, and what we do with the cells is that we embed them in this polysaccharide or hydrogel, and then we research if they can be used as a therapy for arthritis. The reason that we're encapsulating them is because if you do a stem cell treatment or a cellular treatment and you just inject the cells in a patient, they will migrate somewhere else. However, when you put them in a hydrogel, you can maintain them and localize them in a region of interest, and they can remain viable for long periods of time, given that therapeutic efficacy that we are looking for.
Thank you. The greatest challenges. So, what are the biggest challenges you've come across in studying these cells, and how have you overcome them, or how are you planning to overcome them?
So one of the biggest challenges that I have seen and we have seen in the field is donor-to-donor variability. So not everybody has the same cells. Not all cells act the same way. So we will have here many different donors, and we will try to see if they all work at with the same potency. They will have the same therapeutic effects. However, that is not true. Some donors will have cells that are highly efficient, and you can see that they have drastic changes as a therapy, and some others will not work the same. So that is one challenge that we have seen in our lab, and we have also seen in other studies, including clinical studies, where some clinical studies will show the cells were very effective, patients were having less pain, and they were actually having better outcomes. However, you will also see other clinical trials or clinical studies where they will see that there was no effect at all. So another challenge that we have seen is that not all treatments are done the same way. So in our case, we use in vitro, so we use it in the lab, not in animals or patients. We will have cells that are cultured with the same number as the cells that we're trying to treat. So it's like a 1-to-1 ratio of treatment. However, in a patient, you will not have that. You will have your cells that you're injecting, but your patient will have way more cells, many cells that you are trying to affect with this. And cell number, then again, is a big problem. You will have clinical studies where people will use a low cell number, like 1 million cells, and some other studies will have 20 million cells. So how do you draw a line of what is the therapy, how is it effective, and if nobody is comparing the same thing? So that is a big challenge that in the field right now it exists.
Okay. And then, can you talk about a little bit of the achievements that you've found while researching?
So one of our achievements so far is that we have been able to use MSCs and embed them in this hydrogel that is alginate, and see that they have an effect on our cell, our target cell. Then in this case is chondrocytes. Chondrocytes are the only cell that you will find in cartilage, and they're the main responsible cell for building up this cartilage extracellular matrix. So we have seen that when we use our stem cell treatment, you will see better outcomes of decreasing inflammation, and also promoting chondrogenic genes, or genes that will promote the chondrocytes to behave as normal cartilage. But that is just preliminary results right now. So we are working on making them better and more reproducible.
Now, is stem cell research or stem cell treatment, is it available to the public, or widely available to the public? And if not, when do you see that it will be?
So that is an interesting question, because there are clinics right now that are giving stem cell treatments to patients. There is 2 ways to get it. You will get into a clinical trial and receive a stem cell treatment. It could be for osteoarthritis, or it could be for many other diseases. There have been many clinical trials for to treat different diseases. However, you will also see in clinic a doctor will get the stem cells from a patient, and then inject them back into the patient to try to mitigate the symptoms of the disease. Even though that is still being done, it doesn't necessarily mean that it's available for the public, because right now there is no regulation over what the doctor is doing to the patient, and there's no FDA approved treatment right now for stem cells. So in general, right now, actually, this past month the FDA sent 2 letters to stop all practices to 2 different stem cell clinics that were injecting stem cells to the patients that were obtained from their adipose tissue, and they were just taking the stem cells out, injecting them in the patient, and 2 of them they became legally blind after the treatment. So again, we don't know what is happening there. It's not regulated, so it might be available right now at the moment, but the FDA is getting involved to stop and make sure that it's safe and effective. So that might change.
Right. And then, what is the ultimate goal of this project?
So the ultimate goal of this project is that we have a stem cell therapy that we can use for a long period of time to mitigate inflammation in patients that have osteoarthritis and also promote tissue regeneration. That is the ultimate goal of what we're doing here.
That's it.
In the case of this image, the green dots represent the alive stem cells, which is known because the fluorescent dye was able to enter through their membranes. The red dots show the stem cells that are dead because they have no permeable membranes for the green dye to enter through, meaning they are not live anymore. The blue dots show the nuclear stains that bind to every cell, whether it is dead or alive, to show that there is a cell present.
In this capsule, there are approximately 80 to 150 MSCs. This point of view makes the stem cells appear as if they are in a circle, but this is actually an image of them on top of one another.
This image was taken from inside of the Stem Cell Research Lab at Rutgers University Biomedical Engineering School. At the Rutgers Lab, we learned about the type of stem cells that are used to help find an effective treatment for osteoarthritis. In our endeavors, we learned that Rutgers University does no longer use embryonic stem cells to aid in their research projects. In the 1980s, they would use rat and sheep embryonic stem cells to aid in their projects, but due to some controversy, they stopped. Also, due to the fact that when you're harvesting embryonic stem cells, it is very difficult. Also, once you harvest the embryonic stem cells, they are very difficult to use and they do not have a very well mortality rate. So they die very easily and very quickly. So you have to be very careful when you're working with them. So instead, Rutgers University decided to use mesenchymal stem cells, also known as MSCs. The MSCs can be found in human bone marrow or any type of bone marrow, and they're a lot hardier and have a much lower mortality rate. So this is why Rutgers decided to use them. In the process that they use at Rutgers, they will take the MSCs out of the bone marrow of their donor, whatever type of mammal it is, and once they harvest them, they will add pigments, and then they will encapsulate them in an alga medium. And in this alga medium, they can tell with the dye that they added to them before, they look under microscopes, and it's a 3D image that is produced within the microscopes, and they have 3 different colors. They have green, which means that they're alive, they have blue, which means that it's just a nucleus stain, and they have red, which means that they are dead. And that image was shown in a previous slide.